Hello. So today I'm going to be talking about protein NMR and how it is used to propel drug discovery. Now, I realize that people are not all that interested in the technical aspects of NMR, so what I'm going to be showing is real-life examples that will show how NMR is applied strategically, the kinds of results that you sort of typically get, and hopefully I will leave you with some useful takeaways. So I'm going to present some lessons learned from all these studies, and hopefully you'll get some guiding principles that you can apply to future studies. So the examples I'm going to show all involve what most people consider to be the most difficult target class for drug discovery, and that is protein-protein interaction. I'll be talking about finding small molecule modulators of protein-protein interactions. This is an achievement that for a long time was thought to be impossible. And then I'll be also talking a little bit about an approach that's becoming more and more popular, which is the fragment approach, so even smaller modulators of PPIs. And then if time permits, I also want to go to sort of the larger end of small molecules, and that would be peptides, and talk about the peptidomimetic approach as applied to protein-protein interaction. So this slide is just meant to kind of wow you with the large number of protein-protein interactions that are occurring in the cell. We now know that there are about 20,000 proteins in the cell, and almost every protein interacts with at least one or sometimes many other proteins. And it's thought that there may be as many as 600,000 protein-protein interactions happening in the cell. These have evolved, and so they're functionally important. And for any given disease state, it ought to be possible to find a protein-protein interaction as a potential drug discovery target. So if the promise is so great, if you look at the marketed small molecule drugs and ask how do they work, there are very few that work by modulating protein-protein interaction. So if the promise is so great and they're such attractive targets, why are there so few marketed drugs that actually work this way? Well, I think to a large degree the lack of success is due to a lack of trying. So there's kind of a fear that a small molecule will not be able to interfere effectively with a protein-protein interaction. I mean, proteins are big molecules, and the protein-protein interface is a big area. And you don't find a lot of deep indentations in those sorts of interfaces. So affinity is achieved by summing up a lot of weak interactions. And the small molecule, it was thought, would be too small to mimic enough of those interactions to get decent affinity. And people were saying, well, it's like putting a pea down on a football field. It's just not an effective way to go. So I think that just sort of scared people away from even trying one of these in this target class. So when people did try and did screening, it turns out that there's a high false positive rate when you do a screening of a protein-protein interaction. These exposed hydrophobic binding sites can attract opportunists, so small molecules that just kind of glom onto a hydrophobic surface. And when you do your screening and you get the sort of typical hit curve, the most potent hits are usually false positives, so that the authentic hits are lost within the body of the screening curve. Now, for some targets, you can sort of, like kinases, for example, you can look at the hits and you can say, well, this looks like a kinase inhibitor because it mimics ATP, the natural substrate. But for these protein-protein interactions, it's hard to just look at the hits and just visually tell whether they're real or false. And then when people do the screening and they do the hit assessment, it turns out a lot of times you just have no authentic hits at all. So you end up with no leads in your program. And I think the reason for that is that these compound libraries, so the traditional corporate compound libraries that are used for drug screening, don't contain the right kinds of scaffolds, so the scaffolds that are appropriate for inhibiting protein-protein interaction. Selectivity issues is something downstream you might worry about. You know, proteins can have protrusions that will keep away the unwanted partners, but a small molecule really isn't big enough to have something like that. So you worry about promiscuity and toxicity due to non-selectivity. And as I was saying, for a well-behaved, so people were sort of gravitating toward easy drug discovery targets, only to find that there's no such thing as an easy drug discovery target. I mean, kinases, it's easy to get hits, but then you have other problems with toxicity and selectivity. But in terms of lead discovery, for something like a kinase, you can kind of, as I said, look at the hits and say, well, this is real and this is false. But for the PPIs, you have a high false positive rate, and the most potent hits are not real, and it's hard to authenticate the hit. So what's really needed is a method for assessing the hits and pulling out the authentic hits and eliminating the false positives. There are a number of methods that we can do that with, but if those methods also suffer from false positives, then you might do several methods, do a Venn diagram of the results, and hope for a nice intersection where all methods agree that these are real. But a lot of times when you do that, you get a Venn diagram with no intersection. So they're all choosing out their own breed of false positive, but there's nothing authentic. So what we have found is that NMR and X-ray crystallography are sort of the gold standard methods for hit validation. So, especially with X-ray, obviously, if you can see your compound bound to a protein, it's got to be real. NMR is the same thing in solution, and you have your NMR spectrum. You can see the protein, you can see the compound. They're both floating around in there. And if there's no interaction, then you have a pretty reliable indicator of non-binding. So NMR is one of the few techniques where you can get a reliable readout of non-binding. I mean, with crystallography, if you don't get a crystal structure, that can be due to just problems with crystallization. So NMR is a good way to really definitively say this is a false hit. The problem with these two methods is they're protein consumptive and they're slow. So in a project, you have chemists who are eager to get started optimizing hits, and then you have biologists who are saying, well, if the real judge of a lead is if it works, it's real. So I really don't need any sort of biophysical hit assessment. If it's working, it's real. And the chemists are saying, well, if I start modifying my scaffold and I see some SAR, then that's all I need. I really don't need biophysical assurance. But of course, once you've started a chemistry program or a sort of a biology program, it's very resource intensive. So these are very expensive ways of identifying the false positives, and you can waste a lot of time. You would have been better off waiting a little bit for the biophysical characterization. So this is sort of the advertisement slide. So I've been talking about hit validation and assessing binding and determining which hits are authentic binders. And then also with NMR, you get a footprint of where the compound is binding on the protein, which can be very useful. But there are other things we can do if you go to sort of lower resolution methods, just looking at the protein. If you do a quick spectrum of the protein, you can tell whether it's folded properly. If you're trying to compare two batches of protein or a protein with a new construct, you can tell whether you have correct folding. You can do the hit validation incorporated with the screening, so you can just do your primary screening with NMR. It's a little slower than higher throughput methods, but you're simultaneously getting hit validation. So in terms of total time, it might actually be faster to do the screening with NMR from the start. And then if you're willing to wait and you want to achieve higher resolution, NMR is capable of getting full three-dimensional structures of protein and protein-ligand complexes and peptides as well, in the same way that NMR is often used to get structures of small molecules, but in this case you're also getting the three-dimensional fold of the protein. So NMR and X-ray are the two methods by which you can get full 3D structures of protein. And in NMR, you're in solution. So, as I was saying, NMR is good for classifying screening hits. I mean, what everybody wants is a ligand that binds to the active site, binds reversibly, and gives good perturbation, so it's reasonably well inserted into the body of the protein. NMR will tell you, well, with kind of rinse-out experiments, you can ascertain whether your compound is binding covalently or not. That may be a good thing or not, depending on your program. As I said, you get a footprint of the binding site, so if the compound's binding to a different site, that will be revealed by the NMR analysis. And again, that may be a good thing or it may be a bad thing. As I was also saying, NMR is a reliable indicator of non-binding. Also, sometimes you'll see things that bind superficially. So I do see a lot of publications where people have used NMR to verify the binding of hits, and they're showing a very small perturbation, which, in my experience, is not indicative of a good binder or a good lead, certainly for a drug discovery program. So you want to distinguish the good binding from sort of superficial binding and non-binding. Also, because you're observing the physical state of the protein, you can tell whether your small molecule is causing the protein to aggregate or unfold or do something undesirable. So it's funny how many of these small molecules are able to kill a protein. And one danger of using chemistry to decide, sometimes you can optimize a compound and get a better and better protein killer, and you think, oh, I'm getting SAR, I must have a good hit, but you're just kind of, optimizing something that's going to end up toxic and non-selective. Okay, so this is sort of the bread and butter experiment that's used for hit assessment. This is a nitrogen proton two-dimensional NMR spectrum. So every NH along the protein backbone will give a spot in the spectrum at its nitrogen and its proton frequency. So you're really getting a sentinel, sentinels along the entire backbone. So you have nice extensive reporters of what's happening at the protein. And so in black, we have empty protein, and then in red, we've added a ligand, and you can see that in this case, you have a good binder, and you see a lot of chemical shift differences between the free and the bound species. So this is how we ascertain whether you have good binding. And then, as I was saying, because you know where all these NHs are in the backbone, you can map the location of binding. So you look for the biggest perturbations, and then you kind of highlight that region of the protein. So in this case, here's the structure of the protein in cartoon form, and then I've just made sort of a surface for the most perturbed residue. So that immediately tells you what your binding site is. And then if you want, you could try docking in your ligand so that it fits. And in many cases, this sort of footprint plus docking was found to be very close to the actual 3D structure once an X-ray structure of the complex or an NMR structure of the complex was obtained. So this is a fast way of getting some very useful binding information. So as I was saying before in my slide about why it's so hard to find PPI modulators, I was saying the traditional compound libraries don't contain PPI-type scaffolds, so protein-protein inhibitor-type scaffolds. So what does that mean exactly? So over the years, we've actually now had a number of successes on finding drug-like modulators of protein-protein interactions. And from those success stories, we can start the candidates and what represents a PPI-type scaffold. So the one success story I'm going to talk about is the finding inhibitors of the interaction between p53 and NDN2. So just quickly, p53 is a very famous protein. It induces apoptosis, and its levels are controlled by NDN2. So NDN2 binds directly to p53 and kind of induces ubiquitination, which leads to destruction of p53. So NDN2 controls the levels of p53, and some cancer cells evade apoptosis by overexpressing MDM2 and thereby disabling p53. So finding an inhibitor of the MDM2 p53 interaction was thought to be a good approach toward a cancer drug. And so we did a screen of our compound library against this protein-protein interaction. The library was about 1 million compounds at the time, and we did get dozens of hits, and then NMR was used to assess the hits and determine which ones were legitimate, and it turned out that only one of the scaffolds was a legitimate finder and a legitimate hit and a promising hit for this interaction. Now, we weren't able to get X-ray structures early on, so NMR was used with footprinting and then ultimately with detailed 3D structures to guide optimization of the series. And so the initial hit, which was quite weak, sort of triple-digit micromolar, was developed ultimately into this series of compounds, which we call the Nutlins. These are now super potent small molecule inhibitors of p53-MDM2, and they are actually still in clinical trials for leukemia. So the Nutlins, and then we have second and third generation follow-up scaffolds, which are still chugging along in clinical trials. So NMR was a critical technique to sort of discover the leads and then optimize the leads for the series. And just sort of quickly to look at the binding strategy. So here I've overlaid the structure of the Nutlin bound, and then in green I'm showing how p53 binds to NDN2. So p53 achieves affinity by inserting 3 side chains into this NDN2 subpocket. So a leucine, tryptophan, and phenylalanine. And you can see that the Nutlin replicates these 3 interactions, but it does it much more efficiently. So with a protein, to get rigidity, you really need to use a lot of hydrogen bonds. That's really all nature has for achieving rigidity. So you have this helix in order to get these 3 side chains in. The Nutlin can do this much more directly. It achieves rigidity just by having a rigid organic scaffold. And you can see that it is, even though it's mimicking the interactions, it's doing it with different chemotypes. So here, aliphatic being achieved with something aromatic and vice versa. Also, the entry vectors can differ. So here are cases where the Nutlin is coming in perpendicular to the way natural partner does. So in terms of chemotypes and geometry, there's a lot of leeway in terms of being able to duplicate the natural binding with an organic molecule. This is really good news if you're looking for PPI inhibitors, because it means you have a lot of leeway in terms of the chemotypes and scaffolding. So in addition to MDM2 in the cell, there's a protein called NDMX, which would also be nice to inhibit. And so we did another screen of the P53 NDMX interaction. And again, NMR was used to assess the hits and to pull out the authentic hits. And you can see here's this HSQC spectrum where you see perturbation upon binding. So this is indicative of a good deep insertion type binder. And so these inhibitors, interestingly, it turned out to induce dimerization of NDM2. So, I mean, there are a lot of ligands that are known to bring two proteins together, but this was the only case we knew of where two ligands stack together and then bring in two proteins. So here I've sort of taken away the second NDM2 molecule, but left the two inhibitors. So you can see that they stack, and then the phenylalanine, the tryptophan pocket is mimicked by one inhibitor, and then the phenylalanine pocket is mimicked by the second inhibitor molecule. And then you see the same thing symmetric on the other NDM2. So this is a kind of surprising binding strategy. That again, so NMR was indicating even from the beginning that we were getting broadening, suggesting that the dimerization was happening, and eventually we figured out the actual binding strategy. So now, in addition to the NDM2 P53 case, other groups and also the Roche groups have found a number of drug-like protein-protein interaction modulators, small molecules. And if we look at those collectively and we start asking, okay, what attributes do they share? Are there some common attributes that we can say this is a PPI-type scaffold? One thing that jumps out when you look at them collectively is how three-dimensional they appear. So we wanted to ask, is this something that's just sort of phenomenological, or is this real? So we looked for, first of all, cases in the protein database where there's a structure for a marketed drug complex to its target. And it turns out if you go looking, you find 63 of these. And then we collected all the cases where we had a successful drug-like protein-protein inhibitor and an X-ray structure or NMR structure of the inhibitor bound. And we had 25 of those. And then we just basically calculated for the ligand the third dimension, so basically the thickness of the molecule as a measure of the three-dimensionality of the compound. And if you plot those against the typical drug, so in pink are the protein-protein inhibitors, and then in blue are the typical drugs. You see that the protein-protein inhibitors are clearly more sort of three-dimensional than typical drugs. And that three-dimensionality, sometimes it comes from having a rigid scaffold where things are projected into three dimensions. But sometimes it comes from stacking. So a molecule that could normally just be kind of linear decides that upon binding it wants to, or is happier, stack. And so this is sort of a voluntary three-dimensionality. So if you're preparing a protein-protein focused screening library, you may want to consider the propensity of your ligands to stack, because that can be an effective way of binding to the PPI interface. So So then, knowing this, we went and looked at our corporate compound library and found that it was very flat. It was composed of very flat molecules. So we asked, can we construct a PPI-focused or PPI-directed compound library by just collecting three-dimensional scaffolds? So we went and looked at the Cambridge small molecule database of X-ray structures of small molecules. And so one of our templates was the P53 interaction, so trying to mimic in three dimensions the binding motif. And we found scaffolds that in fact provided a good three-dimensional match for those. So these are compounds that are in the database, so somebody made them and they crystallized. So we commissioned the synthesis of a bunch of these to compose basically a PPI-directed library. And in fact, the PPI library did display a higher hit rate against PPI targets, so about an 8-fold higher hit rate than the conventional corporate library. And here's an example of we found a hit against MDM2, sort of a novel scaffold. Still not able to show the structure of it, but the NMR just showing that it's binding to MDM2 and it's binding at the same site as P53. So not only a higher hit rate, but also finding novel scaffolds. And we had other projects where we were finding hits for systems where we had found no hits with the conventional library. So now I do want to say something about the fragment approach, which is an approach that's becoming more and more popular now. So people have done this long enough so that they've pretty much established that for a typical drug, where the molecular weight's usually about 500 or so, a good fragment size is about 150 to 250. So this is a good way of exploring compound space in fragment mode and then eventually getting to something drug-like. But for these PPI inhibitors, where the final molecular weight could be 700 or even higher, is this a good size, or what is a good size for a fragment that would lead to this kind of drug? And so we asked, even though the Nutlins were not discovered with a fragment approach, in retrospect, could we have found the Nutlin scaffold with fragments? So we sort of deconstructed the Nutlin scaffold to answer this. So if you consider the Nutlin, and schematically you think of it as a central template with 4 arms on it, so the P pocket, the Lu pocket, the trip pocket, and then there's always a piece that seems to be sticking out of the active site. Could we have found this via a fragment approach? Well, the individual fragments or the central scaffold don't give significant binding. And if you have 3 arms coming off the scaffold, you do get significant binding. With 2 arms coming off, sometimes you don't get binding, especially in a case where you just have 1 of the subpockets filled, but in a couple cases where you're filling 2 of the subpockets, you do get binding. So this seems to be the smallest fragment of the parent Nutlin that still retains binding affinity. And we were able to get X-ray structures of some of these. So this is just showing the parent Nutlin in white, and it's just kind of reminding you how it overlays with the P53 peptide. And then this is the Nutlin by itself. Now this is a scaffold or fragment rather, with 3 arms on it. It's missing the part that's sticking out in the solution. And you can see it still binds, and in a very similar way to the parent. And also the MDM2 hasn't changed very much. Here's another one with 3 substituents, this time missing the A ring. And in this case, the MDM2 overall hasn't changed much, although this tryptophan has sort of kind of changed position to sort of fill the hole created by the lack of this ring in the Nutlin. But But overall, the MDM2 doesn't change much. And then here is the fragment with two substituents. And again, even it is binding in a very similar way to the parent, with just these two substituents. So this seems to be the smallest fragment that retains decent binding affinity, and it seems to be binding in the same way that it does in the parent. Now, the size of that molecule is a little bit bigger than a conventional fragment. So it's in the range of 300 to 350 rather than 150 to 250. So, as I was saying, we could have discovered enough, and if this compound had been in our fragment library, we screen it, we find a hit, then we start elaborating, we get better affinity, keep elaborating, we could have come up with another that way. So what I was saying was that the fragment being more in the 300 range, if you're building a fragment library targeted against the PPI target, you may want to skew toward the larger end. So here are the key attributes of PPI-type scaffolds. So they seem to be larger than the conventional drugs, more three-dimensional than the conventional drugs, and again, this is sometimes achieved by stacking. They mimic the critical interactions mediated by the natural side chains, but with different chemotypes and with different trajectories. So that's good news in terms of prospectus for finding PPI inhibitors. I didn't harp on this, but we find many times that there are parts that stick out in the solvent. So I was saying that protein-protein interfaces are devoid of deep pockets, they're kind of shallow, but your hits are three-dimensional. So if you're three-dimensional in a shallow pocket, there must be some part that's sticking out, and we do, in fact, find that to be the case. The exact role is unknown, but it seems to be some sort of capping function. So that's the first part of the talk. I think I have enough time to quickly get into the peptidomimetic part. So we're going to switch gears now and start talking about peptide drugs, which are appealing because they have a niche, which is somewhere between the small organic molecules and the kind of protein drugs. So there are systems where you need something that size to get effective modulation. But peptides, of course, they're unstable, they're hard to deliver. So people have spent a lot of time trying to make peptidomimetics, so something that's peptide-like, but also kind of a hybrid between the peptide and an organic molecule to try to improve the properties. And they've tried a number of strategies for doing this, and today I'm going to talk about this one, which is replacing parts of the peptide with our organic scaffold. And of course, that's a risky thing to do because nature, these peptides have evolved to fit properly, and if you start chopping pieces off, you worry that you're going to lose a lot of activity. But one advantage of chopping out a lot of the peptide is that you get a cheaper molecule to manufacture, because making a peptide is expensive and is a function of the length of the peptide. So anything that shortens the peptide should help in terms of cost of goods. So I'm going to be talking about PYY, which is a 36 residue peptide that belongs to sort of the neuropeptide Y family. And PPY was thought to be a good target for weight loss and diabetes. So it was known that if you inject a version of 3 to 36, slightly truncated version of PYY, you get reduced food intake, and this was sort of suggestive that this would be, if you could mimic this with a drug, you'd have a good diabetes and weight loss drug. Also, people who undergo gastric bypass sometimes or often find the anti-diabetic effects happening right away. So even before the weight loss, which is the point of the bypass, they start to see good sugar regulation. And so this has been linked to elevation in PYY levels following the bypass surgery. So our goal was to take the PYY 3 to 36 and make a version that might be longer acting, but with equivalent potency, and something that's novel. So the natural was already sort of taken, so we needed something patentable. So we used NMR to help the design of these peptidomimetics. There was a kind of obscure publication out there that showed that you can remove residues 5 through 24 and with just kind of a linear spacer and still get decent activity. So our goal was to try to replace those residues in kind of a more sort of organic scaffold type way. So we determined the solution structure of the peptide, and surprisingly, it had a structure. So many peptides, when you look at them in solution, they don't have a covalent structure. But this one had a helix, and then the N-terminus came around, and there were interactions showing that this N-terminus gets close to the body of the helix. And then if you look in detail, so if we're cutting out residues 5 through 24, we need to bridge, we still need to keep residues 3 and 4. So we want to bridge this gap, which turned out to be 9 angstroms. So we looked for commercially available organic compounds that were pretty rigid and that spanned a distance similar to what we needed. So we found this collection of commercially available compounds. Then we incorporated them into the peptide where we are removing residues 5 through 24 and just substituting in this organic spacer. And these did have activity, and the best of these was this PQA spacer, which is the most potent. So we, just looking at sort of a 1D proton NMR spectrum of these peptides, you see the version which with kind of a linear spacer isn't changed much from the parent, but with PQA you see a dramatic change in peptide, indicative of some sort of structural inducement. And sure enough, if you do the structure of that peptide, you get a nice coherent structure where the organic spacer is giving a rigid spacing. And then if you compare, so this is just one of the structures, and it shows that this tyrosine seems to be stacking with this organic spacer, which may be helping to keep the structure together. If you overlay the parent peptide, which is in brown, and then this peptidomimetic, which is in green, you see that the peptidomimetic is able to mimic the structure of the parent quite well. So the spacer is doing exactly what it was meant to do. It's kind of a much more efficient trajectory to get from this end of the helix to this N-terminus of the peptide. And sure enough, this peptidomimetic showed activity. So a pegylated, slightly modified version of that peptidomimetic turned out to show good activity. So just kind of to come back and summarize and do the kind of BioNMR advertisement again. So as I was saying, we do a lot of hit validation, but we can also do hit discovery by doing the primary screening. We can do very rapid protein assessment to assess folding, and then if time is appropriate, then we can do full 3D structures of proteins, protein-ligand complexes, and of course peptides, and you just saw the value of having a peptide structure. And I just want to end by thanking my many colleagues. Obviously, a lot of this talk drew from a lot of different projects, a lot of work, and there are a lot of people involved in collaborating. So I thank them, and then I thank you for your attention. titrate in the ligand, then you see the chemical shift, perturbation, and you can, from that, you can derive a KD.
Yeah.
If it's super tight, then it's kind of a with and without. You don't get that progression. So you can say it's tighter than X, but you can't get an actual.
You seem to focus very much on protein-protein interaction, and it looks like a lot of the things you can demonstrate with the NMR is small molecules getting in the way of those interactions, or perhaps making them a bit more sticky in some cases. So that's more of sort of an antagonistic axis. Thinking about the agonistic axis, is there potential, or is there some application for NMR in looking for transduction of signal and confirmation through proteins by a small molecule? Is there anything that can be done to look at wholesale changes, triggered by a small molecule binder?
Yeah. Well, I think if the activity is just a matter of binding, then that should be mimicable. So you may be talking about cases where binding then causes a conformational change to the other side. So because you're monitoring the whole protein, you would see that by NMR. So you would see perturbation at the site, and then you would see perturbation remote, and you would say, oh, it's not involved in binding, but it's a conformational change triggered. If you wanted to mimic that, you could assess whether your hits were mimicking that or not. So at that level, you could. Now, getting to the whole kind of, domino effect, probably not, but at least in that regard.
So that would be a good thing, would you presume, such an overall structural change from just aggregation or something?
Yeah. Well, you can tell from the spectrum whether it's aggregation, whether it's just a nice conformational change or an evil. But yeah, so you can tell whether it's just